Hey everybody, welcome back. It's Miss Chotzi again. Welcome to part 2 of our melanin, DNA, and protein set of notes here. So in the last set, we just reviewed the structure of DNA. And then in this particular set, we're going to talk about how does DNA actually become a protein. In order for that to happen, there needs to be some help. See, the issue is that DNA is a really big molecule, and it cannot leave the nucleus. It can't get out. So we need to somehow get the information from the nucleus to the location where proteins are made. If you remember me saying before, proteins are made in organelles called ribosomes. And ribosomes are found in a couple different places. They float around freely in the cytoplasm. There's also some attached to the endoplasmic reticulum, which I don't know if you remember that from junior high, but you might. So how do we get this genetic information out of the nucleus if it can't leave? Well, we need a helper, and that helper is called RNA. So this part of the notes really talks about what is RNA and what is its job and how does it help build that protein, something like melanin that we're looking for. So first, let's get into some structural things about RNA. RNA stands for ribonucleic acid. Remember that DNA stood for deoxyribonucleic acid. So there's going to be a small difference between the two of them. An additional difference is that instead of being a double helix, RNA is going to be a single helix, so it's only one strand. It is still made up of nucleotides, but the nucleotides are slightly different. The sugar is different. Instead of deoxyribose, it's ribose, so it has a different chemical composition that's going to change some of the bonding that happens, particularly with the bases. The phosphate is still the same as it was before, and bases are mostly the same. We're going to have G and C bonding together again, but this time we're going to have A bonding with a new friend called uracil. There's no thymine in RNA, and that has to do with how many available electrons it has for bonding. Thymine and ribose just will not bond together, so there's a fifth base out there called uracil, which has a weird name. We just call it U. So U will be bonding with A. So anytime you see U's around, you know that the molecule that you're looking at is RNA. So what RNA actually does is it's a temporary molecule. It doesn't exist for very long, but it acts as a messenger to bring information from the nucleus, meaning where the genes are, where the DNA is, to the ribosome where we're actually going to be building that protein. Okay, so let's get into comparing and contrasting what DNA and RNA have in common. So there are some similarities that you see in green here, that they're both made up of repeating nucleotides, they're both very long molecules, and they both contain a sugar, a phosphate, and a base. Now the details of those are what makes them different. Also, things that make them different are locations. DNA is only found in the nucleus, but RNA can be found in multiple locations. It's going to be made in the nucleus, and then it's going to leave, so you'll find it in the cytoplasm as well, and then we'll talk about its connection to ribosomes in just a bit. Next up, the comparison of the two sugars, deoxyribose versus ribose. You don't need to know the details of the chemical compositions of each, just know that they're two different sugars. Double-stranded for DNA, single-stranded for RNA, and then at the bottom here we have our four bases, highlighting the ones that make them unique. So in DNA, A bonds with T, G bonds with C, and in RNA, A bonds with U standing for uracil, and G bonds with C. So remember that if you see U's around, that you know you're looking at RNA and not DNA. So how do we actually make this melanin molecule that we were talking about earlier? Well, remember that melanin is coded by a series of genes, and those genes become proteins called melanin. So we need to somehow bridge the gap from how we have DNA to how we have proteins, and the bridge is going to be the function of RNA. So RNA is going to help us get there. Now, what is a protein exactly kind of plays a role into understanding what RNA actually does. A protein is also a really big molecule. So melanin is a really big molecule, and it's also made up of smaller chains or smaller pieces, just like in DNA. So in DNA, there are nucleotides bonded together, but in proteins, they're made up of amino acids bonded together. Amino acids exist everywhere on Earth. We make them inside of our cells. We also pick them up through our diet. There's about 20 that are really essential for protein creation, or what we call protein synthesis. So I'll show you what some of those are in just a bit. But the way we build a protein is we have to assemble these amino acids into a chain. And that chain is really unique for that particular protein. So no two proteins have the same amino acid chain, or you can think of it as a sequence, having the right amino acids in the right order. There's a two-step process that gets us there. They both sound very similar, transcription and translation. If it helps, transcription is alphabetically first, and it is first in the list as well. So transcribing means you're going to take the DNA and you're going to make a copy of it into a new form called RNA. So you just like make a copy of the gene. And then in translation, you're actually going to build or assemble your protein. Okay, so that's what we mean by transcribing and translating. So transcribing is really just like writing something down in a different language, and then when you translate it, you actually find meaning behind it. So that's where the words transcription and translation come from. We're going to get into those amino acids in just a bit here. But first, I want to talk to you about transcribing, because it's kind of the most important part. Because once a gene is transcribed, it will definitely be built into a protein. It's just a matter of when and where. So transcription begins inside the nucleus because of that. Of course, that's where the gene is. So in the area of the DNA where the gene is, let's say it's melanin, you're going to unwind the DNA. How does it unwind? There are little chemicals, little enzymes in the cell that do the unwinding. So they break apart those weak bonds holding A, T, G, and C together. So forgive my drawing skills here, but let's say that this is the area where our melanin protein is. So we have, oh geez, this is tough. We have all these bases that are going to be separated from the complementary bases on the other side. So we've like opened it, or they sometimes call it unzipping. So you unzip the gene, and then you have all these like naked bases. So you have all these bases that no longer are bound to their friends. So they're lonely and they're looking to bond. So what you will do is we will begin to assemble a complementary RNA molecule off of one side of that double helix. And it is a particular side, but you don't have to know that. It gets way too technical at that point. So you build this little, in blue, here's your little RNA molecule, and then now you basically have made a copy of that gene, but it's just in a slightly different language. The RNA is also built complementary to that DNA. By complementary, we mean the base pairing rules still follow. The type of RNA that we've actually made in this diagram here, if I could add there, is called messenger RNA. And the messenger is exactly what it says. It's going to bring the message or the code outside of the nucleus and deliver it to the ribosomes. It's not going to actually know what it means. It's not going to actually build the protein yet, but it's just simply it gets you from point A to point B in the cell. So if I gave you, here's a skill for you. If I gave you a sequence of DNA, like let's say that this, is part of the sequence for melanin, could you then figure out what's the complementary messenger RNA? So you use your base pairing rules again, but this time note that because you're making RNA, you're not going to be writing any T's. T's will be present in DNA, but not in RNA. So if we have T in DNA, that would pair with A in RNA. A is going to pair with U, its new friend. G and C still go together like normal. Here's another A. A pairs with U in RNA. T pairs with A, C pairs with G, and G pairs with C. So this would be a sequence. So it's in a slightly different language. We've transcribed it. Sometimes this is even called a transcript. When we look at DNA and RNA, we often, it becomes very important to know that we're going to read it in groups of 3. So every 3 bases in messenger RNA is going to mean something. So this little guy is going to get like tweaked a little bit, modified. It's got to be protected as it leaves the nucleus, but it will leave the nucleus through tiny pores, just like pores in your skin, that are small enough for RNA, but way too small for a big molecule like DNA. So hence the reason why RNA exists. So I've got a couple pictures here showing you the basics of transcription. There's 1,000,001 animations online. I'll link some of them to you in our in Google Classroom. But for now, just want you to see what transcription is really all about. This big purple guy here, this is your nucleus. We've got in blue your double-stranded DNA. You're going to make a copy of that in pink. That's your messenger RNA. And then you've got your little opening or pore in the nucleus. That's where it's going to leave the cell. On the right side here, you've just got a more detailed molecule giving you a little perspective to understand that the genes in DNA are kind of continuous along that double helix. We've got 3 genes highlighted with some different colors here and some space in between. So when you transcribe, you're only transcribing a very small part of the DNA. You don't want to code for all the genes in your DNA at once, because then you're going to build proteins that you don't actually need. So if you zoom in on the diagram, they've showed you what the sequence is in DNA here, and then you would build the complementary sequence in RNA. So we've got AAA to begin with, and then the transcript, the RNA, the messenger RNA would be UUU. Now notice they have sequenced these in bases of 3 again, so they've just kind of put some brackets down. Every 3 bases in an RNA is called a codon. It's kind of appropriately named like it's a code. Codons are really important when you want to figure out what amino acid is going to be put in that one particular position. So the amino acid story begins in step 2. So step 1 was all just about making messenger RNA, but step 2, the translation part, is about interpreting what the RNA, the messenger RNA, is telling you. So on the screen here, you've got this big glob here. This is supposed to represent what a protein might look like. Proteins are globular, like big, balls of stuff. Notice there's a lot of twisting in there, and you've got different colors representing different chains. A protein is actually made up of multiple chains of amino acids. Also, FYI, this process is going to happen in the ribosome, but also ribosomes themselves are also made up of RNA, and it's called rRNA. The little r stands for ribosomal, so it's always easy to remember that one. A ribosomal RNA makes ribosomes. Okay, so in translation, what we're going to do is we're going to take amino acids and we're going to string them together to form a protein. And we're going to use the messenger RNA as a tool to tell us which amino acids are going to be put in what order. I mentioned before that the DNA will transcribe into the RNA, which will then be read in bases of 3 called a codon. So on the screen here, you've got some examples of some codons. AUG is a codon, CAG, and AGU. So what we're going to do is we're going to figure out which amino acids those codons are telling us to put in a particular order. And then I'll talk about that little tRNA in just a minute. So how do I know what amino acids I'm supposed to look at? Well, this table is called a codon table, and these are the names of all the 20 amino acids that are used to create proteins in the body. So what you have here, this little, for example, UUU, that's called a codon. Okay, so codons are simply just sequences of 3 bases. And these are all the different ways you can combine A, U, G, and C together. There's actually 64 of them. So there's 64 codons, but there are only 20 amino acids. A lot of them have similar names or suffixes to their names. You've got phenylalanine, leucine, isoleucine, valine. I mean, they all kind of sound the same. There's a couple of oddballs in there. But what you would do is you would look up your messenger RNA sequence in this table and figure out which amino acid it codes for. So back up to our sequence at the top here, we've got AUG as our first codon. So I would then go and look, where is AUG on this chart? And they are in order, if you can figure out here. AUG is right over here. This is probably the most important codon. This is called the start codon. Every gene in the body begins with a start codon. Okay, and it signals the beginning of one gene, and it just basically lets your cells know when the beginning of a gene is versus where the end of a gene is, so you don't accidentally make a gene and a half of another gene put together. So this one is called methionine, and we would use an abbreviation to write that out. We would just write MET. So that is the first amino acid in our story. We would then look up CAG to figure out what the next amino acid is. That one's over here. It's called glutamine. Okay, so we would just abbreviate that. We'll write GLU. And then our next one is AGU. So I would look up AGU. And it is over here. Okay, that one is called serine. Okay, so we'll abbreviate that SER. And most people abbreviate them because they're just way too long to write. Now, in reality, this would go on and on and on, maybe 100 times, maybe 1,000 times. Genes are really big, so therefore the codes that they produce and the proteins they produce are really big as well. Now, when this gene eventually ends, it will end in a very specific way. If you see in the table here, there are three codons that code for stop. Okay, so every gene ends in one of these three codons. That is how the ribosome knows that this is the end of the gene or the end of the amino acid chain, and it's time to release the protein then into the cell, and then the protein is done. So you will actually see that in real life when I give you some longer sequences to practice with. Okay, I'm not forgetting about this little thing called tRNA. We're going to get back to that in just a minute. So I'm going to give you some practice to work on where you have to transcribe and then translate your sequences. So I gave you a little analogy here just to remind you of the significance of mRNA and codons. AUG is the start codon for every gene in the cell, and then there are also stop codons, and there's 3 of them to choose from. And a good analogy of thinking about what messenger RNA is like is that the start codon would be like the capital letter of a sentence. The stop codon would be like the period in that sentence. The sentence itself is information, meaning what protein I'm going to make, like melanin, and then the individual words in the sentence are like amino acids. Okay, so I mentioned before, you saw before that there was this little guy called tRNA up here. So what is tRNA all about? Well, tRNA answers the question, how does the amino acid get to the ribosome in the first place? Like why is it attracted to one particular code and not another code? Well, amino acids are attached to molecules called tRNA. So that's our third and final type of RNA today. T stands for transfer. Its job is to transfer one amino acid into the particular chain of amino acids in a particular spot so that you end up making the right protein that you're trying to make. So this is a picture here of a tRNA molecule. It's a little bit like a clover, a 3-leaf clover. It looks like it's double-stranded, but it's not. So it's a single-stranded RNA that's been folded a bunch of times so that there are some attractions between some of those bases. And at one end here, you're going to see that you have an amino acid attached. This one just happens to be PHE, which stands for phenylalanine. There would be 19 others that you will find. And how does it all work? Well, there's one really important spot on the tRNA molecule. And by the way, it's kind of shaped like a T, so that helps a little bit. And it's right at the bottom here. Okay. At the bottom here, you see the word anticodon. An anticodon is the opposite or the complementary of a codon. So let me backtrack, sorry for moving the screen on you, but how did, CAG up here, how did CAG know that glutamine was the one that was supposed to bond to it? Well, glutamine had a very specific, what we call anticodon. It has a 3-digit code at the bottom here that would then bind to the complementary messenger RNA. So, for example, with phenylalanine up here, the anticodon is GAA, so that must mean that the codon that it bonded to was the complementary, so it would be CUU. So if your messenger RNA said CUU, then you would know that phenylalanine is going to be the only one that connects to it and bonds to it. So another good example here at the bottom, if you had AGC as your codon, your anticodon would be UCG. So it's simply complementary. We're still a type of RNA, so notice there are no T's to be found. So it's just strictly RNA to RNA. So let me backtrack one more time here and finish up this little picture. Sorry if it's making you dizzy. If my methionine up here was AUG, what is methionine attached to? It's actually attached to a tRNA molecule that has a codon, an anticodon that reads UAC. So these two things are actually attached. That's why the methionine goes to the right place. For glutamine, it would be GUC. These two things are attached to each other. For serine, it would be UCA, and these two things are attached. So that's why serine knows that when AGU is present, it's going to find it. Okay, skipping back ahead. Well, skipping forward. So there's a lot of good animations, and I'm going to show you some links to some for translation because it's kind of neat. It's a little bit like a conveyor belt, like a factory. This teal object here, this is the ribosome. Okay, and you've got tons of them in the cell. So these little factories are churning out all over the place because it turns out you have to make a lot of proteins all the time. Here, down here, coming in like a ribbon is your single-stranded messenger RNA. It's going to enter the ribosome kind of like a car wash, like a car moving through a car wash. And all along here, you have all these different sequences, all these different codons. So let me just separate how I think it's going to be separated here. Every 3 bases, I'm drawing a line. I know where to start and end my lines because I'm looking at my tRNA molecules right in the middle here. So here's your like a tRNA molecule. Up at the top here is one of your 20 amino acids. Then we have GCA, that's the anticodon. It would only be attracted to its codon, its complementary codon. So it would only be attracted to CGU. So when CGU pops up in the messenger RNA, then that one amino acid is the only one that's going to be attracted to it. And they do attract. They actually form a bond together. And when that bond is formed, it causes this amino acid to be released. That amino acid will then form a chemical bond with the amino acid next to it. And then you'll just continue to do that until you reach that stop codon, wherever that might be. So it's hard to describe it in a stagnant picture like this, so I will give you some animations to look at. But basically, the tRNAs just pop on, pop off, pop on, pop off. And they start to form their chain of what's going to become a protein. So these are amino acids here. And eventually, when it gets long enough and it combines with other chains, it's going to form a big old protein for the cell. Okay, so a little summary here showing you where translation comes into our story. Translation is the bottom part of our original diagram here, where we're going to build the protein, and it's represented here in purple at the bottom. Once we know our codons and messenger RNA, we can then build what's called a polypeptide. Now, unfortunately, in biology, things always have lots of names. A protein is made up of polypeptides. Polypeptides are chains of amino acids. So, as you notice, methionine, lysine, phenylalanine. The reason sometimes they're called polypeptides is because this bond here that holds any 2 amino acids together is called a peptide bond. And then poly just means many. So, a protein is made up of a bunch of polypeptides. So, there's a lot of really great chemistry words that pop up when talking about protein synthesis, how to make RNA into a protein. Okay, I just want to include one more detailed picture for you about melanin and melanocytes. So notice that melanocytes are not regular skin cells. They're embedded with particular layers of skin. They have these little arms that reach through. So our little melanin is going to be produced here, and then it's going to travel and find other cells to deposit within. Its job is to form a cap around the nucleus. In this case, the nucleus is this little blue dot. And that cap is supposed to help protect them from ultraviolet light. So soon we'll talk about why albinos don't make the melanin. That's where we're going here. Okay, so finally, how do I know if I am understanding anything? This would be an example of a practice problem we would give you on a quiz or a test, if we were to give you a quiz or a test, where if I gave you a sequence of DNA, I might ask you then to find me the messenger RNA, the amino acid, and the tRNA that supports or codes for that particular example. And we'll never give you a full length one because they're probably hundreds, if not thousands of nucleotides long. So we'll just give you a piece. Okay, so the first step is if I know that up top here, this is my DNA, my complementary messenger RNA that would be made is AUG, and that is because the complement of TAC is AUG. Notice the U, because we're in RNA, there's no T's. Okay, then we have CAU, and then we have GUC. And I'm just going to go ahead and kind of split this into groups of 3. It just makes it easier to read, I think. Okay, the next step would be, once I know my messenger RNA, could I find which amino acid? Now, in class, you would have been provided with a copy of the codon table, but it is a few slides previous. But for now, I'll just share with you the answers to be quick about it. If you looked up AUG, once again, that would mean methionine, your start codon. CAU would be histidine, one of our amino acids, and GUC, I'm looking for it on my screen here, is valine. Okay, and remember, in real life, that would go on and on and on to reach a stop codon. Lastly, how do methionine, histidine, and valine know to go where they're supposed to go in the ribosome? They're attached to tRNAs, which are complement to the messengers. So in this case, AUG would be complemented to UAC. CAU would complement GUA, and GUC would be CAG. Oops. And those would then be your anticodons. And remember that these are actually connected together. Okay, so that's everything with transcription, translation, the purpose of RNA. I'll have some practice for you to work on. Hopefully it makes sense.